very well the ethanol production we are executing with the help of the yeast. So the yeast Saccharomyces cerevisiae is used for making the ethanol. Used for making ethanol, very well C2H5OH is nothing but the ethanol. Okay, that ethanol production is happening with the help of the bacteria Saccharomyces cerevisiae. Right now, very well the fermented beverages. Some beverages we are getting with ethanol. Some beverages we are getting without ethanol. Some we are getting with ethanol. Some we are getting without ethanol. Like this, we are not saying. So you need to understand basically. Some are obtained with fractional distillation. Some are obtained without fractional distillation. You know very well the beer, and then the beer is obtained without fractional distillation. Obtained with fractional distillation, whiskey, brandy, gin. Wine also obtained without fractional distillation. So wine and beer are obtained without fractional distillation. Separation of the molecule based on the different boiling point. Separation of the molecule based on the different boiling point. Separation of the molecule based on the different boiling point is nothing but fractional distillation. So by using the different boiling point, we are separating the beverages of various quality. And then these beverages are used for medicinal applications also, that we know very well. You know very well the wine is prepared with the help of the yeast, and that wine and beer is obtained without fractional distillation, right? Now we are entering into the brandy, rum and whiskey are obtained with fractional distillation, right? So in the fermenter, basically the waste products should be removed. Okay, what are the things we have shown here? Can you see this picture here? Okay, the fermenter tank. This is a fermenter tank. Okay, it looks like autoclave. So in the fermenter tank, it is basically attached with temperature control machinery, and also it is circulated with the nutrients and microbes. So this is a fermenter tank, which makes you to understand much more better. Okay, in this fermenter tank only we are doing the microorganism growth, so that microbes of various quality will be producing the different type of beverages, right? So now you were talking about the fermenter plant. Can you see this fermenter plant here? In this fermenter plant, beverages are obtained without fractional distillation. Beverages are obtained with fractional distillation, that we already told to you. And how the fermenter tank is operating, that they're not given in detail in your NCR textbook. So just to make you to understand, this is a fermenter tank. We given this message for you, right? Okay, different parts of the fermenter tank, and each and every fermenter tank, how it works is not there in the CBSE textbook. So we know very well in the whiskey we are getting the components by doing the distillation of the fermented grains, and basically we are getting the brandy in which way we are getting by distillating the fruit juices. Okay, if you're taking about the rum, by fermenting the sugarcane juices and molasses, right? Here, by fermenting the fruit juice, we are getting the brandy, right? So in the fermenter tank, not only the temperature control, not only the pH control, not only the waste product removal control, waste product should be removed properly. Proper oxygen circulation is very important. Proper removal of the waste products, maintaining pH, maintaining temperature, everything is very, very important for making the beverage production at the maximum quality, right? So now we are coming for the next phase explanation is, so I told about fermenter tank, whiskey, brandy. Before going into the antibiotics section, so what I'll be doing is, we'll be going for the related videos whenever you're having time. See this related video in our Go to University portal, and then examples we already had given. In the examples, microbes are used in the production of the antibiotic, includes the key microorganisms and the process involved. We know very well the penicillin is an antibiotic. This penicillin antibiotic is taken from a fungus called Penicillinum notatum. The Penicillinum notatum is a fungus which is used for making the penicillin. Okay, so penicillin is an antibiotic. that is taken from Penicillium notatum fungus. So you can see this particular penicillin is very curative in terms of giving the recovery. Why? Because this penicillin antibiotic is advisable for any age group, and also production is very easy. But the problem with this penicillin production is from the Penicillium notatum is here. This particular penicillin is allergic for few people, so we can't give the penicillin injection for few people. Why? Because it develops allergic reactions in the body, right? So now we are entering into discuss the role of bacteria in the production of biogas. Before going to this biogas production, let me conclude the penicillin is in which way it is more helpful. I already told n number of time to you, this penicillin is resistant, and this penicillin is responsible for killing many gram-positive and gram-negative bacteria. And this penicillin can't be taken orally. We need to give as injection. Why? Because this penicillin antibiotic breaking enzyme is there in the intestine. That particular enzyme name called beta lactamase. So in the CBSE board exam, why I can't take the penicillin antibiotic like this? If they're asking, we need to say beta lactamase is an enzyme. So beta lactamase is an enzyme which is very important for breaking the beta lactam ring or the penicillin. Beta lactam ring or the penicillin. And this beta lactam ring or the penicillin is very effective in giving the curative effect. Discuss the role of the bacteria in the production of the biogas. That topic we are going to discuss. We know very well biogas production is very important, not only for the fuel purpose, and also for running the many engines, not only in running the automobiles, not only for running the super multi-speciality hospitals. We can use for multiple purpose. This biogas plantation is exactly executed with the help of a bacteria called methanogenic bacteria. This methanogen bacteria or methane bacteria, again back I'm repeating, this methanogen is nothing but methane producing bacteria. This methanogen or methane producing bacteria is very important for the production of the biogas. Okay, for the production of the biogas. Anaerobic digestion of the organic material. Anaerobic digestion of the organic material is responsible for the production of the biogas. This we are doing with the help of anaerobic bacteria, which is nothing but methanogen. This methanogen bacteria is very important for the production of the methane gas, which is nothing but a biogas. This we are doing with the help of the cow dung. So you know very well the cow dung is rich in the methanogen bacteria. Okay, cow dung is very rich in the case of methanogen bacteria. And then the second major point, what you need to understand is this methanogen bacteria produces plenty of methane gas and produces carbon dioxide gas. So this methane gas is used as a biogas. This methane gas is used as a biogas, and this biogas is used for cooking and lighting purposes. That is okay. This methane gas production by methanogen is executed in a biogas plant in a controlled pH with zero oxygen limit. That means no oxygen circulation should be there. Why? Because if there is oxygen circulation, this methanogen bacteria can't grow in the biogas plant. And then you need to understand the pH we are maintaining for methanogen should also supported with help of the nutrition circulation. The nutrition circulation in the sense not only the plant debris, and also we need to give the proper amount of water in the nutrition medium for this bacteria to grow more efficiently to produce methane gas. The produced methane gas are collected from the industry exhaust, and then from that particular vessel, whatever we are collecting, this particular methane gas is used for various purposes, right? This is the way we are using the methanogenic bacteria. And next major point, what we need to understand is explain how fertilizers enriches the nutrients quality of the soil. Provide the example of the bacteria and their roles in biofertilizer production. So you know very well the biofertilizers like rhizobium is a bacteria. This rhizobium bacteria is a symbiotic bacteria growing along with the leguminous plants. Growing along with the leguminous plants. This rhizobium bacteria is growing along the leguminous plants. Okay, and this rhizobium bacteria growing along the leguminous plant root having the credential to produce the nitrogen in the available form that we are saying as nitrite and nitrate. So we need to understand nitrite and nitrate are produced in huge volume by using the nitrogen fixing bacteria rhizobium. This rhizobium, while growing along with the root of the leguminous plant, it produces nitrate and nitrate by using atmospheric nitrogen. Atmospheric nitrogen, which is taken by this rhizobium bacteria to make the nitrite and nitrate, which is taken by the plant root very easily. Another example for the nitrogen fixing bacteria, not only azospirillum and even acetobacter. This acetobacter and azospirillum bacteria are growing to the maximum level and making the atmospheric nitrogen. Like this, we are not saying it makes the soil nitrogen. That soil nitrogen is taken from the soil by the bacterial root or plant root. Plant root takes the atmospheric nitrogen. Plant root takes the atmospheric nitrogen? No, it takes the soil nitrogen, which is given in the form of available form nitrite and nitrite, nitrite and nitrate. So basically, this rhizobium alone is not a nitrogen fixing bacteria. Okay, nitrobacter, nitrococcus, okay, rhodospirillum, azospirillum, all the examples we need to give it. If it is 5 mark question, if it is 2 mark, one example or two example is enough. If it is 3 mark, explanation. So we are explaining the answer and then example and then explanation on the example we need to give, right? So why we use the word symbiotic? Why? Because the rhizobium is also getting benefited from the leguminous plant root. That is why we are saying symbiotic association of the rhizobium bacteria along with the leguminous plant root, along with the leguminous plant root. So this leguminous plant root is giving the platform for the rhizobium to survive. This rhizobium makes plenty of root nodules. This rhizobium makes plenty of root nodules over the root nodules of the leguminous plants. Plenty of nitrogen fixing bacteria is growing. Plenty of nitrogen fixing bacteria is growing. Are you getting my point or not? So plenty of nitrogen fixing bacteria is growing along the root nodules. Along the root nodules. Again, back I'm repeating, plenty of nitrogen fixing bacteria are growing along the root nodules of the leguminous plant, and this bacteria does the conversion of atmospheric nitrogen to nitrate, so that what happens exactly, plenty of nitrite and nitrate are formed. This plenty of nitrite and nitrate helps the plant root to take this nitrate and nitrate. This is very very important for not only for making the amino acids and even for making the nitrogen bases. Amino acids are the building blocks of the protein. Nitrogen bases are the building blocks of the DNA. Nitrogen base is also requires nitrogen like amino acid. So this amino acid makes protein. Nitrogen base makes DNA and RNA. For making the DNA and RNA, I required exactly the nitrogen base. For making the nitrogen base, I require the nitrogen. For making the nitrogen, we require the nitrogen in the soil as nitrite and nitrate, which is established by taking the atmospheric nitrogen by the rhizobium, a nitrogen fixing bacteria. This bacteria is growing along the roots of the leguminous plant. This is a message you are understanding. So any one of you are having any doubt, please let me know. So now we discussed about the biofertilizers application by using nitrogen fixing bacteria. And then we told about the biogas production by using the methanogenic bacteria. And then we told about the penicillin antibiotic production by using Penicillium notatum. These are all the repeatedly asked tough questions in the board exam, right? And yeah, the way how they need, in that way we need to present the answer, or else this answer will become a wrong answer, right? And now we become yeah, expert how to write the answer. That is okay, but we need to understand the time limit also. So within the time limit, we need to finish that answer. Accordingly, we need to plan the answer writing. So in the case of biofertilizers, I told not only the rhizobium, Astrobacter, Rhodospirillum, okay, here Clostridium, Klebsiella, these are all the other microorganisms also. We need to give the example if this question is coming as a 5-mark question. Now we are coming for the next phase of explanation is penicillin resists the actually if you are allergic towards penicillin, for those patients we can't give the penicillin. We can give alternative penicillin. Alternative to penicillin is erythromycin, which is not given in the CBSE textbook. So that is why in all the places we are not explaining everything. Erythromycin is an alternative for penicillin, which is not given in the CBSE textbook. So we will be doing only as per the CBSE textbook. So tips for the exam. In this tips for the exam, diagram practice is very important. And second thing is how the microbes are working in each and every sequential process is very, very important. And next process is we need to understand the microbial product, in which way it should be purified and how it should be applied. That also we told you, right? Now, we know very well by using the beneficial microbe, we are getting the high quality product that should be cleaned and preserved properly. So these all things are the part of the tips for the exam, right? So antibiotic is a substance which is formed by one microbe, which kills or control the growth of other microbe. This is the definition of antibiotic. So this which kills uh, which kills other microbes, right? So some antibiotics, antibiotic is a substance formed by one microbe that kills the other microbes. Enough. This antibiotic is totally different from antibody. Many people are getting confusion. So antibiotic is not formed by human being. Only antibodies are formed by human being. You know very well antibodies which are formed by human being are basically of many varieties in the sense not only we used to represent antibody with the symbol capital A small b. Antibody is also called as what? Immunoglobulin. Antibody is also called as immunoglobulin. This antibody or immunoglobulin is a molecule which is formed by B lymphocyte. We know very well the B lymphocyte is a type of WBC which is very, very important for making the antibody. So again, back I'm repeating, antibody is also called as immunoglobulin. This immunoglobulin or antibody is formed by the B lymphocyte. B lymphocyte is a type of white blood cells which is nothing but WBC. So under the B lymphocyte, under the WBC, B lymphocyte is coming. Under the B lymphocyte, immunoglobulin is not coming. B lymphocyte makes immunoglobulin. Immunoglobulin is also called as antibody. This antibody are of many types. This immunoglobulin symbol is Ig. So either you say IgG, either you say IgA, okay, either you say Ig, okay, M, okay, either you say IgE, okay, either you say what IgD. Like this many number of antibodies are there. So IgG, IgA, IgM, IgE, IgD. So these are the 5 different type of antibody. These are the 5 different type of antibody, right? Uh, Now, in this antibody production, is totally different from antibiotics. Antibody is formed by human being, but antibiotic is not formed by human being. And this antibiotic is a substance formed by one microbe, which kills or controls the growth of other microbes. How antibiotic works? Any one of you are having any idea how antibiotic works? Any one of you having any idea how antibiotic? Uh, uh, any one of you are having any doubt? Here, any one of you having any doubt? No one is having the doubt. So in the case of Q&A section, I saw no one raised the question. So I'm going to the next important thing. So some are broad spectrum antibiotic. Some are narrow spectrum antibiotic. What do you mean about broad spectrum antibiotic? What do you mean about narrow spectrum antibiotic? So broad spectrum antibiotic and narrow spectrum antibiotic are the two different type of antibiotic, which is very, very important for making the particular recovery. Broad spectrum antibiotic kills many microbes. Narrow spectrum antibiotic kills only few microbes. Narrow spectrum antibiotic kills only few microbes. Okay, broad spectrum antibiotic kills many microbes. So I already told you, one is narrow spectrum, another is broad spectrum antibiotic, right? And then second thing is antibiotic is formed not by the plant. Antibiotic is not formed by the animal. It is formed only by the microorganism of various type. So this antibiotic is not helpful for controlling the virus. We can control bacteria, we can control fungus, we can control protozoans, but not virus. But not virus. Why? Because many students are writing antibiotic application in the virus that is not acceptable at all. So I can control bacteria, I can control fungus, I can control protozoa. So protozoa, fungus, bacteria, all the three microorganisms can be killed very easily with the help of the antibiotics, but not virus. Why? Because why we can't control virus by using antibiotic? Why? Because virus has both living property and non-living property. Virus has both living and non-living property. So though the virus has both living property and non-living property inside and outside the host. So living property inside the host, non-living property outside the host. Who is exhibiting this? Virus is exhibiting this. As a result, we can't give the effective antibiotic for the virus. Okay. And second thing is, I not told there is no medicine for virus. I told there is no antibiotic for virus control, but there is a medicine for controlling the virus. We used to call it as antiviral chemicals, like you know very well. Antiviral chemicals of various quality and quantity are made in the different places by using the uh, genetically modified microbes. So antibiotics of different quality and quantity we are preparing by using genetically modified microbe. So genetically modified microbe, again back I'm repeating, genetically modified microbe are used for making the industrial products and household products at the maximum limit, right? So genetically modified microbes increase the antibiotic production. Okay, and then we used to call this as transgenic microbe. So this transgenic microbe is nothing but a microbe which can make the plenty of valuable components. Again, back I'm repeating. So transgenic microbe are used for making the uh, components, microbial components. So you need to understand. So microbial components are formed in the maximum limit. Again, back everybody, microbial components are formed at the maximum level uh, by using transgenic microbe, which is nothing but a genetically engineered microbe, right? So some antibiotics will work in acidic medium, some antibiotic will work in alkaline medium, some antibiotic will work in neutral medium. So making antibiotic is not a deal. Preserving antibiotic is not a deal. Okay, we need to make the antibiotic more active in the particular preserving medium. In the particular preserving medium, it should be very, very active. Who should be very, very active? Antibiotic should be very, very active in the particular preserving medium. The particular preserving medium, antibiotic should be very, very active. For example, if we're talking about, so some medium are alkaline like this, I already told you. Some are acid like this, I already told you. So some are neutral pH like this, we told you. So in a particular buffer medium, we need to maintain the antibiotics. Antibiotics. So in alkaline medium, in acidic medium, and in neutral medium, we need to maintain the antibiotics. So you know very well the antibiotics, some antibiotics stops the translation mechanism. What do you mean about translation mechanism? Means protein synthesis process breaking mechanism is called translation mechanism. Transcription mechanism. What do you mean about transcription mechanism? Means the RNA synthesis process is called translation. Protein synthesis process is called translation. Replication process is nothing but a DNA synthesis process. So DNA synthesis process is nothing but a replication process. Okay, RNA synthesis process is nothing but transcription process. Protein synthesis process is nothing but translation process. So each one of this process should be stopped by using the antibiotic, or else it should stop the metabolic activity by blocking certain enzyme activity. So in a metabolism, the enzymes are the specific drivers which drives the metabolism. This is the way the metabolism should go. Okay, sometimes the metabolism should go very fast. Sometimes the metabolism should go slowly. Metabolisms are of two types. One is called as anabolism, and another is called catabolism. One is called as anabolism, another is called catabolism. Okay, so anabolism and catabolism driving enzymes should anabolism and catabolism driving enzymes. Anabolism and catabolism driving enzymes. Anabolism and catabolism driving enzymes are active because of the proper production of the enzyme. Sometimes we can make the enzyme of the anabolism and catabolism inactive by using antibiotics. So there are some antibiotic which stops exclusively certain anabolic enzyme. There are certain antibiotics are there which stops exclusively the catabolic enzyme. There are certain antibiotics are there which stops exclusively the translation of the prokaryote or the translation of the eukaryote. There are certain antibiotic which stops exclusively the transcription of the eukaryote or exclusively the transcription of the prokaryote. There are certain metabolism uh, like DNA metabolism which is stopped by the antibiotics. So the antibiotic, how it works, I already told you. Some antibiotics kills many. In this regard, I am calling that antibiotic as broad spectrum antibiotic. There are certain antibiotic which kills only few bacteria, which is nothing but narrow spectrum antibiotic. Narrow spectrum antibiotic. So again, I am repeating, some are broad spectrum antibiotic, some are narrow spectrum antibiotic. Narrow spectrum antibiotic kills only few microbes. Broad spectrum antibiotic kills many microbes. So this is about the antibiotics. Now we are coming for the next major segment is uh, we are going entering into the industrial products. How the industrial products are formed by using the bacteria, right? So we know very well in the industrial products are so many in number. We already discussed about antibiotic. You know the scientist who discovered the antibiotic is our Alexander Fleming while trying to control the staphylococcus bacteria. Streptococcus is different from staphylococcus. Staphylococcus bacteria is a bacteria which is growing more aggressively. Staphylococcus bacteria is a bacteria which is growing more aggressively. This staphylococcus bacteria growth we can control by penicillin application. The penicillin is discovered by Alexander Fleming for the first time in the world. Okay, here, but the structure was established by two other great scientists called Florey and Chain. So Florey and Chain are the two great scientists who established the structure of the penicillin in detail, along with the penicillin discoverer Alexander Fleming. So here there is a reason why all the three great scientists got the Nobel Prize because of this discovery. Why? Because this penicillin antibiotic we can give not only for the pregnant woman, not only for the newborn baby, for any age group we can give. But only one thing is if they are not allergic towards it. If they are allergic towards it, if they're not allergic towards it, we can give. If they're allergic towards that particular penicillin, if they're allergic towards the penicillin, if they're allergic towards the penicillin, obviously I can't give the medicine. Okay, penicillin, right? But instead of penicillin, as I already told you, we can give erythromycin in the medicine industry. For penicillin alternative, we are using erythromycin, right? This erythromycin is not there in the CBSE textbook. Okay. The cocci, the word indicates many coccus bacteria. So spherical shaped bacteria, Staphylococcus, if it is existing as a cluster grapes, how the grape clusters looks in the same way, if the Staphylococcus bacteria looks, then we are calling that Staphylococcus bacteria as, again back I'm repeating, Staphylococcus bacteria as staphylococci, right? So we need to understand basically the certain antibiotic or resistant or having the anti-infectious property by controlling certain specific metabolism, like this we told you. How the penicillin works, they're not explained in detail in the CBSE textbook. Okay, we know very well in the case of cell wall production of the few bacteria. Cell wall production of the few bacteria is stopped in the sense cellulose of the cell wall. Cellulose of the cell wall production is completely blocked by the penicillin. Penicillin will not allow the cellulose construction in the cell wall of the bacteria. Okay, in the cell wall of the bacteria, right? Now we are coming for the next major point is Staphylococcus. Looks like a grapes, and this Staphylococcus bacteria is causing so many issues. Is okay in the sense what type of issues it causes. It brings the heavy fever. It brings running nose. Okay, you can't be controlled within 1 week. So that is the reason why we are giving penicillin antibiotic. So this is an accidental discovery. We used to call it serendipity, but because he was trying to understand the staphylococcus growth. So at the time he found out in certain place of the nutrition medium, staphylococcus is not growing properly. That is because of the mold. That mold fungus is nothing but penicillinum. So sometime in the board exam they will say mold, sometime they will say fungus, sometime they will say penicillinum notatum, sometime they will say Penicillium notatum, mold, fungus, like this, they will use multiple words. This mold antibiotic penicillin, fungus antibiotic penicillin, okay, and penicillinum antibiotic penicillin is more effective in breaking the cell wall construction of the bacteria by blocking the cellulose synthesis. So we need to understand basically how this particular bacteria is working, right? So now we are entering into the next important concept is once upon a time before the Third World War, okay, the World War II, we know very well, they use this particular antibiotic more effectively, right? So in this antibiotic production has gone up after the after the structure establishment by Flory and Chain. They established the structure in detail about the penicillin. We know very well the penicillin is having a beta-lactam ring. The beta-lactam ring or the penicillin will be broken by human intestinal enzyme. That is the reason why we are giving the uh, insulin injection, but not we are not taking orally. Modified penicillin can be taken orally. The modified penicillin is called what? Ampicillin. The modified penicillin is called what? Ampicillin or amoxicillin. Like this, many varieties of modified penicillins are there. So this modified penicillin can be taken orally, but penicillin directly can't be taken orally. We can give only as an injection. So once upon a time, before 100 years or 50 years back, we can say the penicillin injection be given in less concentration. Penicillin injection we given in less concentration, but nowadays we are giving penicillin injection in the huge concentration. So 2 to 3 ml of the injection, 2 to 3 ml of the injection. In this, once upon a time, we given only 10,000 units. But nowadays, we are not giving 10,000 units. We are giving what? 5 lakh unit. So then only the microorganism will die. So we are not giving 10,000 units. Why? Because our microorganisms develop the resistance in few decades. In the last few decades, develop the resistance against this penicillin antibiotic. So that is the reason why we are giving high concentration of the penicillin with specific reference to units number in the ml of the injection. Now we need to understand basically the NS chain and then the Flory are the two great scientists, along with our Alexander Fleming discovered the penicillin, and all three got the Nobel Prize. So many people, instead of writing Alexander Fleming, they are writing William Fleming. Don't write William Fleming, because William Fleming discovered mitosis process, but not penicillin, right? So here, if you're talking about roles in the disease treatment, so the antibiotic Kali Hansa, whooping cough. So the cough will be not stopped. It will be continuing it or it is getting prolonged for 2 to 3 minutes. So diphtheria is called Kalagotu. Okay, here leprosy is called what? Kush Rog. Kushta Rog, we will say in South India, in North India, Kush Rog. Okay, in the case of North India, we will say Gal Gotu. Okay, Gal Gotu. Okay, here in South India, Gal Gotu. Okay, here Kali Hansa. Okay, okay, these are the different type of diseases. Okay, here we can control by using the antibiotics. The antibiotics of various quality and quantity are available in the market with different preservative agents. For certain age groups, certain antibiotics can't be given. For example, if you're talking about strong antibiotics like amoxicillin ampicillin I can't give for newborn baby. Okay, whereas I can give for the grown up, right? And second thing is, if you're talking about the antibiotics are having a wide range of bacterial infection. So the world heavily relies on antibiotic for treating a wide range of antibiotic infection. You know very well the antibiotics treatment is very, very important. Why? Because our immune system alone can't perform the defense mechanism. It needs the supporting factors, antibiotics, and we need to give the antibiotic in such a way the normal cell should not go for any type of damage, and also our normal cell activity should not go for any damage. So normal activity of the cell should not be disturbed. In a such a way, we need to give the antibiotic. For example, if I'm giving strong antibiotic to any age group, they'll have a diarrhea. So I need to give the multivitamin and multimineral supplements along with the diarrhea. Not along with the diarrhea, along with the antibiotics. So along with the antibiotics, what I need to give? Multivitamin, multimineral tablet. Along with the antibiotic, I need to give the multivitamin, multimineral. Okay, why? Because the strong antibiotic is having a negative impact. Okay, it brings diarrhea, and also it kills beneficial microbes of the intestine. You know very well beneficial microbes of the intestine, not only E. coli, Lactobacillus lacti, also is responsible for the production of the high quality water soluble vitamins and the fat soluble vitamins in the intestine. So this will support our intestine digestion absorption mechanism. See, without the help of the vitamin B12, iron can't be absorbed. For example, I ate 5 kg of iron. Not even a single gram of iron will be absorbed if vitamin B12 is not there. So vitamin B12 is mandatory in the intestine for iron absorption. Vitamin K is very, very important for making the blood, for making the hemorrhage, uh, hemorrhage restriction, or else we can say the clotting restriction will be taken care by vitamin K, right? So this is a message we need to understand. So one is we once upon a time when we are making the fresh fruit juice, a lot of flushes will be there in the case of fresh fruit juices. That flushes are digested with the help of the microorganism enzyme. So now next basic information is here Aspergillus niger is a bacteria. Aspergillus niger is a bacteria? No, it is a fungus that is used for producing citric acid. Of course, we require citric acid production. Why? Because the citric acid is used for various medicinal applications. Astrobacter aceti is a bacteria which is very, very important for making the acetic acid. Okay, you know very well the citric acid is very, very important for making the bitter taste production. For example, I need to make some products which is having the bitter taste. For this, I'll be using Aspergillus niger. By using Astrobacter aceti, we can make acetic acid production. By making Clostridium botulinum, we can make butyric acid production. We can make butyric acid production. By using the Lactobacillus lacti, we can make lactic acid production. For by making the yeast, we can produce ethanol. We can produce ethanol, right? So we know very well the ethanol production, how it is happening. By using the yeast Saccharomyces cerevisiae, the ethanol is produced in a huge volume with the help of the fermentation application that we discussed recently. Okay, we know very well the acetic acid is required for making the buffer to maintain the pH of various quality. Why? Because to run the electrophoresis process, which is a process which is used for separating the particle based on the charges. So particles are separated based on the charges. Particles are separated based on the charges by using electrophoresis. By using electrophoresis, right? Electrophoresis is executed with the help of the buffer. Electrophoresis is executed with the help of the buffer. Electrophoresis is executed with the help of the buffer. Are you getting my point or not? So now what we need to understand is basically the acetic acid is used for making the buffer preparation. Like this I told, but acetic acid alone is not sufficient. I need to make sodium acetate also. So sodium acetate and acetic acid is used as a buffer for making the buffer solution. I required acetic acid. Acetic acid is produced with the help of Astrobacter aceti. So what we need to understand basically is the uh, here Clostridium butyricum is a bacteria which is used for making butyric acid, which is used for making the butyric acid. Butyric acid is very, very important for making the many varieties of components. Okay, many varieties of components in the sense what exactly? You know very well the effective neurotransmitter, which is nothing but the which is nothing but yeah. Any one of you can say the answer for this? GABA. GABA aminobutyric acid. GABA aminobutyric acid is a neurotransmitter for making this I-liquid butyric acid. Acetic acid for making buffer. Citric acid for making butter. Butter? No, not butter. Bitter tablet. For making the bitter tablet, we require citric acid. And for making the vitamin C in the lab, we are using the citric acid. Okay, and for making the various drinks, we are using citric acid. Citric acid can be consumed very easily after making the proper dilution. We know very well by using lactobacillus lacti, we are making the lactic acid. This lactic acid is very, very important in the curd for maintaining the proper acidity. Proper acidity in the sense we don't want huge acidity, we don't want um, the medium acidity also in intestine as well as in the esophagus. We need acidity in the stomach, which is already given by HCl in the stomach. That we don't want. We need only the lactic acid is very, very important. Lactic acid is very, very important um for making the components a little bit acidic. Why? Because lactic acid is organic acid. Okay, we can use lactic acid in many food products. That food products rich in lactic acid gives a proper light acidic condition. So organic acid, right? So we need to understand basically the ethanol production is possible with the help of the yeast Saccharomyces cerevisiae we discussed now only, right? So Lactobacillus delbrueckii is a commercial strain which is not given in the CBSE textbook. That is why we not told them. Aspergillus niger is a fungus. So sometime in the board exam they will say what type of commercial product we are getting exclusively with the acid producers category microorganism. Acid producer category microorganism like fungus. If you are looking into this, Aspergillus niger is an amazing answer. Astobacter acid is an amazing answer. Clostridium butyricum is an amazing answer. So like this, what we need to ensure the proper answer script making process. So now we are entering into the next important segment, which is nothing but lipase for the detergent. You know very well, lipase is very, very important for digesting the oil spills. Once upon a time, there was a great scientist called Anand Chakraborty. He used genetically modified, we used to call Anand CH. So this Anand CH or Anand C is very, very important for developing the genetically engineered lipase making bacteria. The genetically engineered lipase making bacteria, we used to call that as a very good example for that as Pseudomonas. You know very well the Pseudomonas is a bacteria which is used more efficiently to remove the oil spills. So genetically engineered Pseudomonas is designed to produce plenty of lipase enzyme. This plenty of lipase enzyme makes the oil spill breakage so that the oil filled cloth will be cleaned. Okay, now we are coming for the juice clarification. So if you're talking about the juice clarification, bottled juice purchased in the market are clear compared to the homemade juice. Why? Because the bottled juice, to this we are adding the microbial enzyme. One is called as pectinase, another is proteases enzyme. So pectinases and proteinase enzyme is very, very important for breaking the pectin and protein. So proteins and pectins are broken. Pectinase enzyme breaks the pectin, protease enzyme breaks the protein. Okay, so here this juice which is rich in the fruit flesh, which is digested completely by the application of pectinase enzyme and protease enzyme. We all know very well the plant fruit cell wall is made up of pectin. So the pectin of the fruit cell wall is broken very easily by the pectinase application. And then second thing is, if you're talking about the old plant cell boundaries, okay, old cell boundaries, okay, old plant cell boundaries, okay, are rich in the protein. The protein is also broken with the help of proteases enzyme. So what we need to understand basically, uh, by using proteases, I can break the microbial boundary and plant boundary. By using proteases, I can make all the organisms boundaries, because all the organisms are having cell membrane. So all the organisms are having cell membrane. So cell membrane breaking enzyme proteases we can make it by using genetically engineered microorganism at the maximum level. Pectinase is an enzyme which is used only for breaking the plant cell wall. For breaking the plant cell wall, only I'm using the pectinases enzyme, right? So by using these two enzymes, we can make the clear fruit juice, okay? And that fruit juice will be served with the proper preservatives after proper preservation at the temperature and pH, etc.